Okay, I have my microphone on all the time now.
Yes. I'm going to switch my microphone off now.
Okay, here we go. So to really understand the functioning of an electronic device, you must understand the functioning of the individual components that make up that device, resistors, capacitors, transistors, etc. But you must also understand how they are connected together in a circuit. That is, you must have its wiring diagram. The same is true for the brain. To understand its algorithms, we must understand the functioning of its many types of neurons and the molecular processes which occur at the synaptic connections. But we must also know the details of how the neurons are precisely connected together to form circuits capable of specific tasks. Even seeing the brain's individual wires, the axonal and dendritic processes of neurons, is difficult since they can shrink down to 40 nanometers in diameter, 10 times smaller than the wavelength of light. And these tiny neuronal processes are tortuously interwoven. Okay, I see somebody else arrive. And these tiny neuronal processes are tortuously interwoven in 3 dimensions like a plate of the finest spaghetti. Only 3D volume electron microscopy has the power to image such circuitry. Now, people might think that 3D volume electron microscopy was just invented a couple of years ago, but it actually has a very long history dating back to the 1950s. And I want to do that justice here. So the transmission electron microscope, the TEM, was actually invented in the 1930s and was quickly developed into an instrument capable of nanometer resolution. It took until the early 1950s before a method was invented which could prepare brain tissue and slice it so thin, 50 nanometers or less, so that the details of individual neuronal processes and synapses could be imaged in the TEM. As early as 1958, researchers had begun to apply this ability to thinly sectioned brain tissue for viewing in the TEM to create 3D volume reconstructions of the synaptic connections between neurons. And this image on the right here is actually a 3D reconstruction without a computer. It's actually was made by tracing onto plastic sheets and cutting out those plastic sheets.
Excuse me to interrupt, Ken.
Sure.
To interrupt, I think your view graphs have disappeared from the screen. Can you just reproject it on the screen?
I'm sorry, my...
Let me do that.
I'm sorry,
we don't see the view graphs on the screen anymore.
Okay. I see that.
Just come help you.
Okay. See that.
Right. Huh?
Are you going to do that, Giulio, or should I do it? Are you going to do that, Giulio, or should I?
If you use the menu and reproject it on the screen. Because for some reason, I'm going to do that. You're using this one. You're using the left one, right? Okay.
Yes. I'm using the right one. And I should have just projected it.
Okay, do we have it on the screen back?
There you go. Yes. Yes.
Okay.
Okay, I take that I'm back online here. And microphone is on, so I'm echoing. Okay. So as early as 1958, researchers had begun to apply this ability to thinly sectioned brain tissue for viewing in the TEM to create 3D volume reconstructions of the synaptic connections between neurons. And ever since then, the neuroscience community has relied on this method of serial section TEM reconstruction to study the precise details of synaptic connections between neurons. Because this is a very important technique, and because all of the other techniques that I'll be describing are using very similar technology, especially on the tissue processing side, I'm going to take a moment to go through this technique in more detail. So this is the traditional procedure for serial section TEM reconstruction. An animal is first perfused with glutaraldehyde. The glutaraldehyde is a molecule that has two reactive groups and thus strongly cross-links all the proteins together, locking them in place and preventing them from being leached out in the later tissue processing steps. The brain is then surgically removed and stored in fresh glutaraldehyde overnight, and then the brain is vibratomed to extract a slice, usually less than 100 microns thick, that contains the area of interest. This vibratome slice is then immersed in a solution of osmium tetroxide. Osmium tetroxide fixes the lipids together and prevents them from being leached out in later processing steps. Osmium penetrates the tissue very slowly and can overfix the tissue if left on too long, and this is why vibratome slice thickness is usually limited to about 100 microns thick, although some researchers have been able to get much thicker than that. The tissue slice is then immersed in uranyl acetate, which in block stains the tissue with uranium to provide additional image contrast in the electron microscope. The tissue is then put through a graded series of alcohols to remove all the water from within the cells. And then the tissue is put through a series of ethanol organic solvent mixtures, which removes the alcohol, the ethanol, and replaces it with an organic solvent, typically propylene oxide. This organic solvent is necessary to dissolve the plastic resin to allow it to infiltrate the cells. Because of this organic solvent, you can see why it's important to cross-link the proteins and lipids together. Otherwise, they'd be extracted. The tissue is now put through a series of plastic resin organic solvent mixtures. EPON is the typical embedding plastic that is used because it's low viscosity and heat curable, and it stands up well to the electron beam. And the tissue is placed in a 60 degrees Celsius oven for about a day. And the tissue at that point is then a perfect fossil. Every fine dendritic process, every synaptic connection, and every last synaptic vesicle is preserved for eternity in a hard block of plastic, as you can see here. The tissue is then loaded in a chuck of an ultramicrotome, which is equipped with a diamond knife, and trimmed so that that only a tiny part of the tissue will be cut, typically about 100 microns by 100 microns. The diamond knife of the ultramicrotome is incredibly sharp, about 2 nanometer radius at the tip, and it is fixed in a boat containing water that wets the back surface of the knife. In this way, each 50 nanometer section floats on the water surface tension, eliminating any friction. Christian Harris, a world expert in this TEM reconstruction technique, has produced a set of educational videos of the rest of the process, and I will use stills of these videos here. This is a growing ribbon of ultra-thin sections floating on the water attached to the edge of the diamond knife. Once a ribbon of several hundred sections is cut, an eyelash is used to gently break the ribbon and attach a dozen or so sections. And then a 3 millimeter diameter copper TEM slot grid covered with an extremely thin plastic film, less than 50 nanometers thick, is picked up with forceps. And this grid is submerged in the water and the floating sections are scooped up on the surface of the thin film. These slot grids are then stained with heavy metals, for example lead and lead citrate, and loaded into a TEM. In the transmission electron microscope, the sections need to be held on this gossamer thin substrate to allow electrons to pass through the sample. The grid and the sections are put into the beam of the TEM for imaging. And the result is a series of TEM images that are then aligned in a computer and manually traced to produce 3D reconstructions of neuronal processes and synapses. Unfortunately, the incredible fragility of such ultra-thin sections has made it impossible to volume image and reconstruct large volumes of brain tissue using this TEM approach. The largest reconstructions have been limited to about 0.01 cubic millimeters in volumes, which means that in almost all cases one cannot see whole circuits but only tiny subsets. Here's a typical reconstruction put in context of the whole mouse brain. So this is from a research paper out of Stephen Harris's lab. At the top here is a mouse brain, 15 millimeters long. A tiny section of the hippocampus was removed, put through this tissue processing, and then an even smaller region of that was cut. And from that, a series of electron microscopes was taken, images were taken, and an even smaller region of that was eventually reconstructed. So you can see that this is a really tiny region that has been reconstructed from the mouse brain. And this is typical of these studies since the 1950s. This is like seeing only a tiny part of a large circuit diagram. It's very hard to get the whole circuit when only seeing a part of it. If one could use scanning electron microscopy, SEM, instead of transmission electron microscopy, then one could in theory do away with the need to cut and collect fragile ultra-thin sections, allowing for automation and larger volumes. Unfortunately, until the invention of the high brightness field emission electron source in the 1990s, it was impractical to take high resolution SEM images of biological tissue. There's a wonderful review paper on just when scanning electron microscopes became high resolution. In 2004, Winfried Dink and Horstman showed that this reliance on the TEM for tracing neural circuits could in fact be overcome. They published a seminal paper demonstrating the serial block phase scanning electron microscope method, which could robustly automate the process of obtaining a series of electron micrographs from a block of neural tissue using one of the new high brightness field emission SEMs equipped with a low energy backscatter electron detector. In the serial block phase method, a plastic embedded block of brain tissue is mounted directly in the vacuum chamber of the SEM, and a diamond knife is used to scrape the top 30 nanometers off of its surface. The electron beam then images this freshly revealed surface, sensing the electrons that bounce back off the heavy metal stained parts of the tissue. The diamond knife then scrapes off another 30 nanometers, and the process repeats itself, creating a series of images. This FibSim technique was recently used to image a 350 by 300 by 60 micron volume of the mouse retina that had previously been functionally imaged. And this allowed the researchers to see the synaptic connectivity patterns underlying the recorded functional responses. It's a very groundbreaking work. Following the invention of this serial block phase SEM by Dink, two other techniques for automating the acquisition of 3D volume electron micrographs of neural circuits were introduced. The tape-to-SEM technique and the Fib-SEM technique, both of which also depend on the ability to do high-resolution surface imaging using today's field emission SEMs. I'll first describe the tape-to-SEM technique that I and my colleagues at Harvard University have invented. I will point out some of its advantages, and I'll point out some of its limitations as well. I will then describe the FibSim technique, which has a complementary set of advantages and limitations. And finally, I will propose a new technique which is really a hybrid between these two, which is designed to overcome the limitations of both while retaining both of their advantages. It's a hybrid that can make possible a Connectome Observatory of the mouse brain. So first, the tape to SEM technique. So this is a simple tape collection mechanism that I built at Harvard University to automate the collection of ultra-thin sections for imaging in the scanning electron microscope. This tape collection mechanism is designed to be attached to a conventional ultramicrotome, making the collection of ultra-thin sections automated. It starts with a reel of carbon-coated Kapton film, about 8 millimeters wide, 50 microns thick. That is sent through a bottom pinch drive around a tensioner mechanism, and crucially, it goes around a submerged conveyor belt head that is submerged in the water of the diamond knife of the ultramicrotome. Sections are collected on this conveyor belt, which is drawn up with a top pinch drive and then finally collected on a take-up reel. Zooming in on this tissue collection process, this is the diamond knife with the filled water boat. This is the plastic embedded tissue block. And this is a diagram of an ultra-thin section being collected by the conveyor belt of Kapton film. And this is an image of an ultra-thin section being collected in this way. Usually I'd have a video here, but I wasn't able to convert it. It's a very large video. Runs can last 24 hours and longer, sometimes much longer, even a week, resulting in tens of meters of tape containing thousands of sections. This long tape is then cut into strips and adhered to silicon wafers. The wafers are put into the chamber of the scanning electron microscope for imaging, and the sections are SEM imaged similar to the Serial block face SEM by recording backscattered electrons, and recently we've been using secondary electrons to produce that are produced by the stained parts of the tissue sections. So this is so these wafers are about, which means that you can pack an awful lot of sections onto them. This is an SEM montage of a full wafer containing a bunch of sections. If we zoom in on one of these little sections, we see that it's 3 millimeters long and over 1 millimeter wide. These are huge sections compared to regular TEM cut and collected sections. The advantage of having it in the SEM is we can zoom in on any of these sections and any place in these sections to very high resolution. So if I zoom in on this little box, I can start to see cell bodies. By the way, this is a slice through the cerebral cortex of a mouse. So we're going to we zoom in. We start to see these little white dots are cell bodies. Zoom in to that red box, and we start to see individual dendritic processes and axonal processes. And if we zoom in again, we can see synapses and synaptic vesicles. And I'm going to try to switch to the video here that shows a stack. Let's see if this works. Is that actually working? People see that move?
Oh, yes, it is.
Great, great. I was wondering if anybody was hearing me, actually. I was wondering if anybody... Okay. And of course, so that was just 100 sections, and it probably went by relatively fast. But if you take thousands of these sections, align them, and display them in 3 dimensions, you can, of course, trace individual neuronal processes right down to synaptic connections like you see right here. So using this atom and a commercial ultramicrotome, volumes approaching a cubic millimeter can be automatically cut and collected in just a few days. This creates what I call an ultra-thin section library consisting of dozens of wafers, each with over 100 ultra-thin sections. I've written a custom SEM control software that allows a researcher to direct the SEM to automatically image any subvolume within this larger cut volume. So this software is shown here, interfaces directly to the SEM, automating all the steps of the imaging process. This is the graphical user interface of the software, showing a wafer that has been automatically mapped, where all the sections have been found and automatically numbered by the software. As an example, this is one wafer, 171 sections, from a run of 8,000 sections of mouse cerebellum cut by my colleague Josh Morgan. These wafers are mapped and imaged automatically using the wafer mapper software. And to give you an example, this image shows 16 of these wafers from this one run, containing on just this page of 2,637 sections. The software automatically acquires a low-resolution overview image of each of these sections and aligns these into an overview stack. It's a low-resolution overview stack, but it allows you to then graphically define on top of this low-resolution overview. You can zoom through the low-resolution overview, and then you can graphically define on top of this a high-resolution an area to do high-resolution imaging. So zooming in on that graphically defined area, you can see that we've defined in the software that we're going to take a 3x5 montage of this particular region all the way down through all of these wafers, and elsewhere in the software we defined that we'd wanted it to be taken at 4 nanometer pixel size, at 10 megahertz, percentage overlaps, things like that. And once defined, the software controls the microscope stage in the imaging to acquire the high-resolution montage images automatically. To give you an idea of the timing of this montage is 250 by 150 microns. It was acquired with 4 nanometer size at 10 megahertz rate. To acquire such a high-resolution montage over each section in just this one wafer, which is 166 sections, in just this one region that is shown here, took 40 hours of straight SEM imaging and resulted in about 0.5 terabyte of imaging data. The tape-to-SEM technique has the significant advantage of allowing large volumes to be cut and low-resolution imaged quickly, for example, in a couple of weeks. Then directed random access high-resolution imaging can be performed on any sub-volume of the cut volume. It also allows for parallel imaging using multiple SEMs. These are important points because imaging a full cubic millimeter on a single SEM at the optimal resolution for tracing neural tissue, which in this case would be about 5 by 30 nanometers for this tape-to-SEM process, would require 5 to 10 years, even at the fast rate of 10 million pixels per second. So a serious limitation of this tape-to-SEM approach is the fact that cutting thousands of ultra-thin sections is still a difficult and error-prone process, especially for sections on the order of 40 nanometers and below. The ones you just saw were 30 nanometer sections, by the way. It requires optimal tissue processing and a perfectly sharp diamond knife. The optimal resolution for tracing neural circuitry, that is the optimal thickness, would be even thinner, on the order of about 10 nanometers or below. But this does not seem possible using physical sectioning and collection like this. Such high-resolution sectioning has been achieved, however, using the focused ion beam SEM technique, which I'll go to next. So in 2008, Graham Knott introduced the focused ion beam scanning electron microscope, FIB-SIM technique, for tracing neural circuits. In this technique, a focused beam of gallium ions is used to ablate away a thin layer of the block surface. Now, this is a block that is plastic embedded block, just like I showed in the TEM, and just like we use. A scanning electron microscope is then used to image the freshly revealed surface. This is repeated over and over again to create a 3D volume image. Figure B here shows a block of plastic embedded brain tissue whose surface has been fib milled, and C shows that milled surface imaged by the electron beam. And zooming up, this is an image zooming in on the fib milled surface at high resolution. You can see that the electron images are more than sufficiently high resolution to trace the neural processes in the synapses. Recently, Graham Knott has demonstrated that this technique can achieve resolutions approaching 5 by 5 by 5 nanometers. It's really stunning images. Actually, there's an online journal of visual experiments that's listed here, video of the FIB-SEM process, and I recommend anybody that is interested take a look at it. It's very, very fascinating. So this FIB-SEM technique beats all others in fundamental resolution because it uses a tightly focused beam of ions to slice tissue instead of a diamond knife. Such high resolution should allow extremely reliable, fully automated computer tracing of neuronal processes and automated synapse identification. Such computer automated tracing is an absolute necessity for any truly large-scale connectomics project. So the fact that this has more than sufficient resolution is actually very important. It's definitely the case that in 40 nanometer thick sections or 30 nanometer thick sections you can trace everything within these sections. But if you have even better resolution down to 10 nanometers and below, it makes the computer algorithms for tracing these circuits much, much easier. Unfortunately, this FIB-SEM technique has a fundamental Achilles heel. Due to the physics of ion milling, the highest resolution imaging can only be applied to volumes smaller than 100 microns across. In fact, a more optimal size for the FIB-SEM technique would be 20 microns across. You can imagine why this would be the case, because you've got this ion beam that is being focused only over a short distance. And if you want to take off just 10 nanometers, just 5 nanometers off the surface of a very large block, it becomes impossible. Small block, it's all right. So the key point is, if one could devise a way to losslessly subdivide a larger block of tissue into such optimally sized subblocks, then this FIB-SEM technique could theoretically be applied to arbitrarily large volumes, limited only by the number of imaging machines run in parallel. Such lossless subdivision probably cannot be performed prior to plastic embedding, for example at the vibrotomy stage, since sectioning soft, wet, unembedded tissue seems to destroy several microns of fine neuropil surrounding the cut. Of course, one can losslessly subdivide plastic embedded tissue by ultra-thin sectioning it, although that, as I said, is not particularly reliable because it's so thin. Even if you were to jump to thicker sections, let's say 500 nanometers, though, they are too thin for practical FIB-SEM addressability. A more optimal block size or minimal efficiently imageable volume for the FIB-SEM would be a 20 by 20 micron cube. Let me pause for a second, see if I can explain this. So we have this technique, the focused ion beam SEM technique, which is able to get the highest isotropic resolution, possibly even 5 by 5 by 5 nanometer resolution. And yet it can only achieve these resolutions on things on the order of 20 microns across. If you could divide a brain into blocks losslessly that could go into those, into a focused ion beam, then you would be able to apply this technique to arbitrarily large volumes. Yes, you can losslessly subdivide very thinly, but that is impractical for loading into the focused ion beam because you'd have many pieces of tissue and you'd only end up imaging a very small region before you had to load the next one in. So there's an optimal size for this for subdivision, and I'm claiming here that that's about 20 micron by 20 micron by 20 micron cube, roughly in that area. It's an open question whether one can losslessly subdivide a plastic embedded block of brain tissue into subblocks of this thickness, this 20,000 nanometer thickness, while still being able to trace the finest neuronal processes between subblocks. Attempting to section a standard plastic embedded block at 20 micron thickness results in extreme forces on the diamond knife, internal section due to stress as it bends, and a complete loss of water location. McGee-Russell showed that one could alleviate these cutting stresses by heating the knife to temporarily and locally soften the plastic during sectioning. However, they used a crude metal knife and did not attempt to evaluate the surface damage of these thick-cut sections. What I've been doing is I've been experimenting with modifications to this hot knife microtomy technique to see if it can be used to produce optimally sized blocks for the FibSim imaging, which have such high-quality surfaces that even the finest neuropil down to the nanometer scale can be traced between these big subblocks. So after a lot of trial and error, which I don't have time to go into, I have found a set of parameters that allows for such lossless sectioning of plastic embedded brain tissue. And I'm going to show you these right now. In general, it requires a heated, ultrasonically vibrating diamond knife lubricated with oil instead of water. So this is the heater element I built to heat an ultrasonic vibrating diamond knife. And this is the setup. You can see the this ultrasonic vibrating diamond knife is sitting here in a regular ultramicrotome, and its boat is filled with oil instead of water. So I'll now present the results of a set of experiments that I performed to demonstrate that the surfaces of these 20 micron thick hot knife cut sections are undamaged using this procedure, and show that the fine neuronal processes can be traced from one thick cut section to another. To evaluate surface quality, I will be comparing the matched faces made by a single cut. For the particular cutting event depicted here, this is the section side face, and this is the block side face. So these two faces should, of course, be matched. To compare these matched faces, we would image the appropriate surfaces of these two sections that I'm pointing to in the arrow, with the arrows. And these sections are laid face up and coated with carbon for SEM imaging. So the procedure is a plastic embedded block of mouse cortex tissues mounted on an ultramicrotome sectioned at 20,000 nanometer thickness, 20 micron thickness, using an ultrasonic knife whose boat is filled with cutting oil and heated to 50 degrees Celsius. The retrieved sections are flattened on a hot plate and the oil is removed by dishwashing liquid. The matched faces of the two 20 micron thick hot knife sections are placed face up and imaged in the SEM. So these are the images of the matched faces in backscatter electron mode. And you can notice how they are mirror images of each other, as they should be, because they are coming from the same cutting event. These are images of the on the right here. These are images of the matched faces using secondary electron detection, which is an imaging technique that would clearly show surface damage if it was present. I've taken the slides out that actually show surface damage in previous experiments, but trust me on this, there is no surface damage shown in these images. This will become clearer as we zoom in, Above are the backscatter images. Below are the secondary electron images. Again, no surface damage. Zooming in again at 2000x, no surface damage. Zooming in again, no surface damage. And if I mirror reverse one of these and align it, it can demonstrate that it is easy to trace medium-sized dendritic processes from one to the other. So you might note that these images were taken with a 6 kilovolt beam in backscatter mode. What that means is that there is significant depth seen. The electron beam was high enough energy so that it would see up to a micron down in the block face on either side. And this is why the images change so much as one flips between them. Unfortunately, I had to use such a high voltage for these tests because of the particular tester I was using. Using a different SEM, I was able to obtain lower voltage secondary images on a different set of thick cut sections. Fortunately, these are a little lower quality than what you would usually expect, but they show they do show that you can trace neuronal processes from one to the other. So this is these images are from a low voltage secondary imaging of matte 20 micron thick hot knife sections. So here's the left face and the mirror reverse right face. Zooming in, left face, right face, and coloring the different processes, you can see that it's easy to trace very small processes. Scale bar here is 1 micron. You can easily trace the finest neuronal processes from one of these thick cut sections to the other. Just let me see if I got a question here. Yes, I think what you might be seeing is a little contamination from the oil. Absolutely true. As a final test, I did an atom reconstruction across the matched hot knife faces. So again, the procedure was cutting a piece of plastic embedded mouse cortex tissue using at 20 thickness using an ultrasonic diamond knife. These sections were then re-embedded in plastic so that they could be sectioned at 50 nanometer thickness and collected using the atom tape collection mechanism. I cut a few hundred sections that went through both of these matching faces, and I did a reconstruction on either side of this matched cut. And long story short, the reconstructions were able to trace fine neuronal processes from one side of the face back to the other and back again with no problem. So these results demonstrate that you can losslessly subdivide a block of brain tissue into 20 micron thick slabs. I'm sorry, let me yeah, so I got a question here about what it is that exactly limits the focused ion beam SEM technique to a 20 micron or so volume. If you look at a focus, so if a beam of gallium ions is focused electrostatically, so that it comes to about a 10 nanometer beam size, and that highly concentrated beam is then scraped across the surface of the block to ablate the top few nanometers of that block. And so what you really want to do this milling is you want a uniform high intensity ion beam that is scanned just at a grazing level across the top surface of one of these plastic blocks. Now, That's easy to do if it's a small block, because the region at which this beam is 10 nanometers in diameter is pretty small. If you try to make a 10 nanometer wide ion beam that's, let's say, 1 millimeter in length, it's impossible, because the ions actually push each other away. The focusing optics become very difficult to make. And so what you get is you get a depth of focus issue. Just like in a regular light microscope, it has a limited depth of focus in which you have a high intensity concentrated ion beam to do the milling. And so if you were to try the focused ion beam technique on a block that was a millimeter wide, what you would get is the leading edge of the block being etched at a different rate as the middle of the block, and the end of the block would not be etched at all. And it causes streaking effects and all sorts of things like that. So hopefully that answered the question. Yes. Okay. So these results that I just showed you demonstrate that you can losslessly subdivide a block of brain tissue into these 20 micron thick slabs. This may seem like a trivial result, but I think it may have great ramifications to the field of connectomics. This is because thick sectioning should be robustly automatable. I'll show you how in a moment, to allow 100% reliable sectioning of volumes the size of even a whole mouse brain into a physical library of subblocks of optimal size for random access FibSim imaging. So let me go directly to that. Okay. So recall first that the FibSim surface ablation technique for these optimally sized subblocks can achieve ultimate voxel resolutions as small as 5 by 5 by 5 nanometers, making computer automated tracing very reliable. This, combined with the hot knife lossless subdivision, means that this ultimate voxel resolution and fully automated computer traceability could be potentially applied to trace neuronal processes across arbitrarily large volumes, even tracing a small unmyelinated axon from one side of a mouse brain to the other. So I'm going to finally show you this hybrid of the tape-to-SEM and FibSim technique, and show how this might be able to Connectome Observatory for the mouse brain. So here's a preliminary CAD design of an automated hot knife thick sectioning machine for the mouse brain. So the idea is that this would be able to section a whole mouse brain with essentially 100% reliability, because in a reasonable amount of time, because it's being sectioned so thick. So we start with a precision Z stage. And on top of this sits an air-bearing linear stage with 100 millimeter travel range. And on top of this is mounted a plastic-embedded mouse brain. The mouse brain is set up for sagittal sectioning, as shown here. We add to the hot knife mount, and zooming in on this, we can see how the linear stage moves the mouse brain past the hot knife for sectioning. This is a diamond knife that would be heated. The knife assembly is designed to be very similar to the one that I use, the ultrasonic setup that I currently used for the experiments that I just showed you. It has a piezo vibrator with a counterweight. The diamond knife is 8 millimeters wide. It has a heater element, etc. And of course, there is an oil applicator to lubricate the knife during sectioning. In this machine, collection is tape-based, similar to my Atom design that I showed you. There's a stock of heat-sealable PET film, 20 micron thick, with an outer side that has a heat-activated adhesive. This tape is fed around a tape applicator head, which heats up to 120 degrees C for a few seconds when the mouse brain block is pressed up against the tape, and then cools to set the adhesive. And during the cutting process, a series of 20 micron thick brain slabs are collected, adhered to the tape. and are collected on a final take-up reel. So zooming in on the main action here, this is the heat applicator head that bonds the tape to the block face prior to sectioning. If I remove that, you can see how the tape is attached directly to the block face. And of course, there's this air and water jet for cleaning the oil off the block face. So I'm going to walk through the basic operation steps here. I'm going to remove the heat applicator head in one of the tape guides so that the operation steps are clearer. So here we go. The Z stage presses the block against the tape. The heat applicator head raises the temperature to 120 degrees C for a few seconds, which sets the adhesive. And then it lowers the temperature back down, which sets the adhesive. The block and the tape then translate together because they're stuck together at this point. And the tape is actively pulled up by the take-up reel. So the result is a 20 micron thick brain slab that is securely adhered to the collection tape. The block retracts. The air and water jet clean oil from the block face. And the process repeats. And the result of this is that the entire mouse brain is reduced to a tape, single tape, containing 500 slabs, each from 20 microns thick. This is not a big number because 20 microns is pretty large. At this point in the process, each of these 500 slab faces would be SEM surface imaged to create a set of reference images, which would be needed later to determine sub-block neighborhood relations. And then a person manually adheres dummy blocks, plastic dummy blocks, behind each of the 500 slabs. And these 500 slab blocks are then manually trimmed of free tape. Then each slab block is put into the original machine, and this is a crucial point here. Each of these slab blocks, 500 of them, are put into the original machine for another round of sectioning, this time orthogonal to the original. During sectioning of these slab blocks, the tape is offset so that it does not touch the original brain tissue. I'm going to zoom in on that. So you can see that here's this 20 micron thick brain slab that is being sectioned orthogonally and collected by the tape. And it's offset by about 0.5 millimeter. The result of this second round of sectioning of a slab block is a tape containing 300 tissue pillars, each securely adhered to the tape edge. If we zoom in on one of them, you can see that each of these pillars is 20 microns by 20 microns by the length of the mouse brain, in this case 15 millimeters. This tissue pillar here, I'm pointing to it, but I guess you guys can't see it. This tissue pillar is the mouse brain tissue. We zoom in again. You can see its cross-section is 20 microns by 20 microns. This pillar tape is designed to be optimally sized for high-resolution FibSim imaging. It is also optimally sized to allow volume post-staining with hot ethanolic heavy metal stains, which have been shown to penetrate at least 15 microns into plastic-embedded blocks. This staining issue is very important if we want the fastest imaging rates. At this point in the process, each of these 300 pillars on this tape, and remember there's 500 of these tapes, would be SEM surface imaged on all exposed faces to again create a set of reference images. And this pillar tape is then permanently installed in its own tape cassette with built-in reel motors and a clamp, which can securely hold one pillar in place for imaging while not obscuring its sides from the focused ion beam milling. To allow random access FibSim, there's some redesign that has to go on in the traditional focused ion beam machine. So I'm showing this diagrammatically here. First of all, the beams should be rearranged to be about 90-degree angles to each other. This arrangement allows the focused ion beam to be dumped into a scintillator screen, which is looked at by a CCD with high-powered optics. This would greatly assist setup by first allowing a quick determination of fib stigmation and focus by looking at the shape of the spot on the scintillator screen, and more importantly, to allow quick determination of the block height at which fib milling begins by watching for dimming of the spot on the scintillator. So remember that to mill off 5 nanometers off the top surface of a block of tissue, you have to be very precise for where the focused ion beam is relative to that block. With this slight redesign of the FibSim arrangement, a pillar tape cassette should now be able to be quickly and automatically loaded to image exactly the desired spot on the tape. So this is shown here. A coarse stage movement would be used to load the particular tape cassette into the FibSim. And fine piezo-controlled stage movements would be used for final imaging and fib milling positioning. FibSim imaging of a particular part of the tissue pillar can now commence. And this is showing, so that in purple here is the focused ion beam that is grazing the surface of this little tissue pillar, and in red the electron beam is coming down to image that freshly revealed surface. And on either side here are regions that have been previously volume imaged. Because the entire mouse brain would be divided up across 500 such pillar tapes, this system would allow any 20 micron subvolume of the mouse brain to be addressed by 1, picking the correct tape cassette out of the 500 that have been created, 2, reeling the tape to the proper position and clamping the pillar in place, 3, loading the tape cassette into the FibSim, 4, fine positioning the pillar in X and Y based on matching SEM surface images seen to reference images, and then raising the pillar slowly until the fib align optics records that surface milling has begun. And of course, such random access system would allow for parallel imaging using multiple FibSims. Okay, so finally, let's go through some numbers here. This is, after all, a system that is yet to be built. So what you could do with it if you did have the proper funding to build it. So given the proper hot ethanolic post-staining of pillars and an optimized low-voltage backscatter detector, it's very reasonable to assume that FibSim imaging could achieve at least a 5 megahertz imaging rate, 5 million pixels per second. At 5 by 5 by 10 nanometer voxel size, which should be more than sufficient to allow perfectly reliable computer automated tracing, this would put the image 20 by 20 by 20 micron cube at about 1.8 hours. To account for ion ablation time and automated setup time, let's say that one such 20 micron cube would in fact take a total of 3 hours to image. Now, assume that instead of a single FibSim, that we actually had 20 of these devices operating in parallel. Because remember, lossless subdivision has made this parallel imaging possible. Such a 20 FibSim system would have a total throughput rate of one of these 20 micron cubes, every 9 minutes. Or to put this in another perspective, such a system would be able to image over a 10-year operational period over 500,000 cubes, and each of these cubes could be distributed in any fashion desired throughout the entire volume of the mouse brain. Now, I'm about to answer Giulio's question here. Well, what could one do with such a system which can direct the highest resolution imaging in an entire mouse brain with essentially 100% reliability? Well, if all the cubes were devoted to a single dense reconstruction, that volume would be 4.7 cubic millimeters, which is much less than the size of a mouse brain. It's about 100 times less. So that may be a little bit discouraging, but I don't think we should really consider that as discouraging, because researchers could instead decide to strategically distribute these cubes by imaging 50 separate brain regions within the mouse brain, each with a dense reconstruction of 300 by 300 by 300 microns, and trace over 8 meters of the finest projecting unmyelinated axons between these 50 regions. And if you had 2,000 machines, you could definitely get the whole mouse brain this way. Absolutely, we're going there. But not in this talk. So such a strategy, this imaging multiple separate brain regions, would produce fantastically detailed dense reconstructions of 50 separate brain regions, while at the same time putting each of these dense reconstructions in the context of each other and the rest of the brain, by allowing dozens of fine long-distance axons to be traced into and out of each of the reconstructions, all within the same brain. So, analogous to an expensive state-of-the-art astronomical observatory, and to give you an idea, this system would cost on the order of $10 million and up. For this Connectome Observatory of the mouse brain, we're talking about a small fraction of the cost of a astronomical observatory. If you wanted to do this for a human brain, you would get into the cost level of an astronomical observatory in the $100 million or $200 million range. And I think this thick sectioning makes that very possible. So analogous to an expensive state-of-the-art astronomical observatory, such a multi-FibSim system should not be viewed in the context of a single lab or a single project. Instead, it should be viewed as a potential resource to be shared by the entire neuroscience community, on which individual researchers can schedule imaging time for their projects exploring the mouse brain and, of course, other brain circuits at their finest level. In short, we could refer to such a system as a Connectome Observatory of the mouse brain. And that's all. I want to thank Juan Carlos Tapia for doing the tissue processing for all the hot knife sectioning. Josh Morgan, who cut the cerebellum tissue that I showed you before and processed it. Richard Shellac did all of has helped me greatly with the electron microscope, maintains our electron microscopes, and cut the mouse cortex section. Daniel Berger did a lot of work on imaging those mouse cortex sections, and David Hillebrand helped with the thick cut sectioning, and of course Jeff Lichtman, who is the head of the lab that made sure that all of this research occurred. So thanks a lot. I'll try to catch up with my many questions that I've been ignoring.
Thank you very much. That was really great talk, especially the end. Now, I suggest those who want to ask a question to Swap, just to switch the microphone on one after the other. I'm going to ask the first question. Now, you mentioned a resolution of 5 to 10 cubic nanometers per cube. First, is that correct?
So I switched my mic onto press mode. So yes, the resolution that Graham Knott has shown for the FibSim is 5 by 5 by 5 nanometer resolution. If you look closely at the images, the Z resolution is actually blurred, and that's because of the depth of penetration of the electron beam. So
Okay,
I'm
so
more
there's just
comfortable
one
with
thing.
saying that it's a 10 by 5 by 5.
Great, there is just one thing that I want to know. Is this resolution good enough for doing what we here really want to do? Or has it to be still improved by one order of magnitude?
Can you remind me what we really want to do? I
What
think we
we
know,
really
but
want to What we really want to do is first to do that for a human instead of a mouse. And well, my brain, I hope, is bigger than the brain of a mouse. And second, what we want to do is to read and store this information in such a way as to be able to reconstruct a person after the information. I think that is the end point which we want to achieve. So do you think we have already achieved your technique to require resolution?
First of all, I wouldn't say it's my technique. It was invented by Graham Knott, so I need to be careful about that.
Oh, sorry, Graham.
But the focused ion beam imaging technique. But the answer would be, in my opinion, absolutely yes. This is more than sufficient resolution to determine all the connectivity and the properties of the synapse that are needed to to explain its functionality of the circuit.
Thank you very much.
That's right. Of course, I can't say that with 100% certainty. That's why I prefaced it with, in my opinion. I can go into as much detail on why I have that opinion as necessary, but it might be better to suggest where it might not be sufficient. Okay, so the question is about the property of the shapes you're imaging, and can you do immunostaining? Can you elaborate just a little bit? I'm sorry. On the property of the shapes.
So I'll just switch on. Excuse me. I'll just switch on the microphone. That should be better. I mean, the resolution, it's certainly amazing. The question is, you're trying to build a simulation, a numerical model, and we don't have models which work yet. So the problem is you are inferring the properties of the shapes to put into the simulation. But all you're seeing is heavy metal stained voxels. Is that enough or not? I mean, there is no way to tell until after you've done it. Okay.
Yeah, I agree with that. I would say that there is already evidence that the knowledge of circuit connectivity is reliably correlated with the function that is recorded in cells. So, for instance, the Winfried-Dink paper that just came out this year that did a reconstruction of cells in the retina showed that you could reliably predict the functional responses of those cells based upon the physical synapses that they were making with other cells. So there is evidence of that type. Certainly, it is not 100% evidence that this would be able to work, but there's a complicated answer to this, and let me go a little bit into that. So, if you look at the types of neural circuits that have been proposed in theoretical neuroscience for how different regions of the brain work. So, for instance, the visual cortical circuits for picking out different visual features in the environment, those theoretical models are contingent on the synaptic connectivity between neurons. And you would expect from those models that if you knew the synaptic connectivity between collections of neurons in the visual cortex, that you would be able to reliably predict their receptive field responses. Same thing is true for models of hippocampal memory. If the hippocampus is storing memories in the form of stable attractor states, like a Hopfield network, you would expect to be able to determine those stable attractor states based upon the connectivity of the neurons that can be seen structurally. Does that answer the question a little bit? So let's see. So can you see the synapse type? So I would argue that yes, you can see the synapse type, as you can see if it's type 1 synapse, type 2 synapse, things like that. You can see the size of the synapse. But more importantly, what people usually mean by synapse type is they see a single electron micrograph which shows a piece of a synapse, and they say, gee, I know that that's a synapse that's contacting this particular neuron. I want to know if it's excitatory. I want to know if it's inhibitory. I want to say if it has this neurotransmitter or this neurotransmitter, what is it? And that's a very difficult question if you only have a single electron micrograph. But I'm not talking about having a single electron micrograph. I'm talking about a system where you could literally follow that synapse, follow the axon that is synapsing on a particular target cell, follow that axon back to its cell body so that you would know for sure that this came from this region of the brain, and therefore it must be a cell, a cholinergic cell, or it must be an excitatory cell, or it must be a cell of this type, and that would give you all the information you need to know for synapse type without immunostaining. So I'm looking at Anders' question. Let me put that on the stack. So in your opinion, that information would be enough. So in my opinion, that information would be enough. Not at our current level of understanding of the brain. If somebody handed the entire, if somebody handed us a 5 by 5 by 5 nanometer voxel resolution image of an entire human brain today, we would not have sufficient neuroscience knowledge to interpret that in order to simulate it. But given the knowledge of the next 50 years of neuroscience, I am, in my opinion, very confident that we would be able to take that same 5 by 5 by 5 nanometer voxel resolution of the human brain and simulate it and get the same behavior in the same person. So I'm looking at Anders' question. How well is the tracing and scan interpretation software developed, in my opinion? So I just went to a conference at Genelia Farm Research Campus, and there were a bunch of talks on the state of the art of tracing software. It's certainly not 100% perfect, but it is quickly developing. It is an area where the software people have said, wow, we're finally getting quality images that we can work with, and they're developing a lot of good algorithms for this. Without a doubt, the focused ion beam data that was floating around at the conference was the most exciting for the people developing software for tracing neural circuitry, because it makes the software much more reliable. So I've seen software that can automatically trace neuronal processes throughout this data, The reliability estimates are they're a little bit hard to interpret. Let's put it that way. A harder question is determination of synapses. But I've seen other papers that actually identify all the synapses in a block of tissue that's focused ion beamed. So in my opinion, it's coming quite nicely along the software development.
No other questions, I think? Anyone want to ask something else? Let's see in the tech chat. Eugen.
Okay, go ahead. Sample preparation.
Yeah, I think I'll do it in audio because it would be too much to type. So in this case, you're working with mice, and you're doing perfusion with crotaldehyde through the vascular system, and then you're soaking it. And also, you're soaking it after dividing it in the. Assuming a human sample, you would have to perfuse through the vascular system, but you cannot diffuse it through by soaking. So you have to work actually. Well, you could section, but in this case, you want to section it after embedding. And so the question is, how are you going to prepare it, especially how you're going to embed it with resin, with polymer?
Yeah, that's a great question. Actually, at the Society for Neuroscience conference right now, there is a poster being presented by a guy named Sean Mikula, who's, I guess, famous for the brainmaps.org site. He is now working in Winfried Dink's lab and has been developing techniques for preparing an entire mouse brain for electron microscopy serial block face imaging. He's just now presenting this in poster form, but it looks like very exciting work. It's not completely done yet, but he has been getting very reasonable results by simply tricks a mouse brain in osmium tetroxide, and then doing the rest of the plastic bedding steps and dehydration steps by simple immersion with very, very long time periods. Also, the people down in at Texas A&M, Louise Abbott, and have been doing plastic embedding of whole mouse brains by simple immersion. I personally think that if you're trying to go, so if you're trying to get a mouse brain prepared for something like this, you might be able to do it even without perfusion of osmium. And you definitely can get it to work via perfusion of osmium and having the later plastic embedding steps simply done by immersion. I've seen enough evidence to convince me that that's going to work for a mouse brain. For a human brain, I think it might be more tricky. The vascular perfusion with glutaraldehyde followed by osmium should work for a human being as well as it does for a mouse. The plastic embeddings, the dehydration and the plastic embedding steps, I really don't have any good information on whether that could work for such a large scale. I think there are tricks around it where you could use the ventricular system and the subdural space, and really use the cerebral spinal fluid areas as another route to get the dehydration and the plastic embedding to work. And I should mention that this is why I have a prize out there for showing that you can preserve a whole brain, a whole large mammalian brain by any method. let me just see if I did I adequately answer that question? And then I look and I see that Mike Perry has a question here.
Hello?
Yes,
Hello, can anybody hear me?
I can hear you.
You can
is this
hear
Mike
me.
Perry?
It's Mike Perry. Hello. My question, if you can't read it, is in cryonics, we use cryopreservation to preserve the brain. And do you think there's any better way to do it today in terms of wanting to map the brain at some future date? Is cryopreservation the best you can do, or anything else better, in your opinion?
Yeah, it's a great question. I don't think of it as better or worse. I think there's advantages and disadvantages to cryopreservation versus chemical preservation. The first thing I would say is I am not convinced of the adequacy of preservation of the current cryopreservation techniques. I still need to see better electron micrograph data of it. It's not awful, but it is. I would obviously still like to see more. The chemical preservation techniques, there's even less that have been done at the human scale on that. So it's really difficult to say that in any meaningful way that would be better. Because it's really hard to compare the two. There's just not enough data out there, and
So
that I've seen.
you just don't know what would be better based on what we know now.
That's right. That's exactly right.
Okay.
And my standard answer to this is that more research needs to be done. And but in terms of mapping brain tissue, as opposed to bringing somebody back. In terms of mapping brain tissue, like the type of Connectome Observatory stuff that I just talked about, chemical preservation is really the way to go with today's technology. If you're going to be using electron microscopy to do high-resolution maps of neural tissue, it is very, very difficult to get high-resolution images of cryopreserved vitrified brain tissue. People have done focused ion beam imaging of vitrified tissue samples, but the lack of membrane staining and such that you get in the chemical preservation techniques means images are much worse, and it's a much more difficult technique. So if we take the idea of actually bringing people back, which off the table, which of course I don't want to take off the table, but if we were to take that off the table, just look from a scientific perspective on what would be the best way to preserve a brain for scientific research of this particular type that we're interested in mapping circuits of the brain, there's no question that chemical preservation would be the best way.
Okay.
So I'm looking at some of the questions. Oh, I'm sorry, I'm slow.
Yeah, that is a question. Well, in fact, it's a point that Mike made on the Alcohol magazine a few months ago. So I understand completely your point of view, and that's the point of view of a scientist. You're saying that more research is needed, and we should not really see anything more at this moment. But suppose I have 6 months to live, then I know that more research is needed, but I don't want to hear that. What I want to hear is whether I, in these conditions, could conceivably be able to take advantage of what has been done in the field of chemical brain preservation.
Yeah, I think that's a great question, and I would agree with Eugene here that vitrification would be your best shot, without a doubt, today. because there is no chemical brain preservation that is available for a human being right now, except dumping in a vat of formaldehyde, which I don't think is really a viable technique for long-term preservation.
Okay, let me ask you the difficult question then. Do you think that the current research in chemical preservation could advance to the point that brain preservation services for human could be offered in a time frame of until the end of the decade?
Absolutely. I mean, this is what's so frustrating to me, and I'm sure everybody else in the cryonics community, and just actually the scientists that have signed up to be advisors on the Brain Preservation Foundation. Most of them, in fact, I think all of them are not signed up for cryonics, but they want something like that to be available. That's high quality. If there was really a concerted effort to develop either or both of these technologies or something else for brain preservation, it would be easy to have a hospital procedure that is high reliability, ready to go in every hospital if we wanted to, well before the end of the decade. It's all a matter of will, and there is no will, apparently.
Thank you very much. That's a great question. And what I would say hearing that is let's go and try to create this world, wouldn't you?
Yeah. Oh, and I would be very interested in that ultrastructure preservation of fossilized insects literature. If you can send it to me, that'd be great. Eugene.
Okay, I believe there are no more questions. Let me take a last look at the text chat. Somebody, which is Dmitry, is saying that Penrose and Hameroff told about microtubules carcass neurons, which in superposition can contain consciousness. Can Connectome Observatory help in modeling these microtubules superposition?
Yeah, I probably should wave off of this question because I haven't really looked into their proposal. But I think the Connectome Observatory probably wouldn't have much to say in terms of this. Although I will point out that the resolution of the FibSim technique and even these other techniques is capable of tracing microtubules, just the gross structure of such. And Anders says, let's see, I get the impression that you think most aspects of the scanning is very scalable. Scalable in size, yes, I agree. Let me double check that I understand what you mean by that. So given a small amount of funding, let's say a couple $1 million, all of this stuff could be demonstrated on a mouse brain with a single focused ion beam machine that's set up to do the random access imaging. At that point, you would be able to say, well, gee, if I had 100 of these machines, then I could, I would be running 100 times faster. So yes, if there was a Manhattan project or an Apollo project devoted to mapping a human brain, it would still take, well over a decade, I'm sure, but the technology we can already see. And of course, there's other technology that may completely wipe this out. If nanotechnology and nanoassemblers come to fruition sometime soon, then that might offer a totally different route to achieving these high-resolution structural maps of the brain. But right now, I think it's very credible to point to the existing technology that we can already see is really able to do it with if there was sufficient funding for scaling. So our microscopes are in ultra-high vacuum. We are not, so some of the original Cira blockface SEM papers used low vacuum to prevent charging. The particular imaging that we do with our thin sectioning on wafers is able to be done in high vacuum because we're collecting on carbon, so there's no charging. The FibSim imaging is also done in high vacuum. Or ultra high vacuum. Yeah, there is a paper on this. It was actually Richard Schlack, one of my colleagues, did vitrification, did focused ion beam on vitrified tissue. So I could look up that reference and send it to you. The images were not of the quality that you expect in chemical preservation, but it was the high vacuum did not really present a problem, if I remember correctly.
Any more questions? Okay, no more questions at the moment, and we don't want to keep Ken beyond lunchtime. Can I ask you something unrelated, Ken? How could you make the animations in your PowerPoint in JPEG format? I was really wondering about that.
Yeah, well, I use a CAD software called SolidWorks, and it has animation built into it. I use that animation sometimes, and sometimes I just did screen captures. But really, it's SolidWorks that I use for most of these.
Ah, that's great. Can I ask you to send me the original PowerPoint? We're going to write about this. And maybe
I'll see what I can do.
The best to do is to put it on some place like a slide share.
Yeah, I have to double check on some of these images if they can be put out online in that way. But yeah, I'll double check.
Okay, thank you very much, Ken. That was really a great talk.
Well, thank you. Thanks for everybody that came.
And I hope the money comes to be able to, doing all these things for real and for human beings before the end of the decade, because some of us here are getting really old and we could use something against that. Let's hope for.
Yes, absolutely.
Okay, I'm now going.
Yes, and thank you so much for inviting me.
Thank you so much for coming. Okay, I wish now to collect all the video files that have been recorded. One is Eugen, another is